Hello everyone and welcome to Instant Biology by Dr. Nila. In today's lecture, we would be discussing about pBR322 vectors. Now you would be knowing that we have already started a series talking about the different types of vectors, and in the previous lecture we had discussed about the plasmids. So what are the salient features of the plasmid that we had discussed? We have already discussed about this particular vector pBR322, but that particular video was in Hindi. But in this particular lecture, I have decided to talk in English about the PBR322 plasmid. What are the features of PBR322 plasmid? What are the characteristic features? Apart from that, what is the construction of PBR322 plasmid? What are the genes that are present on PBR322 plasmids? Apart from that, we would also understand the methodology through which we can perform the how PBR322 plasmid is utilized in the genetic engineering. Basically, that also we would be studying in this lecture. After that, I would also be sharing some of the previous year problems or questions that are commonly associated with PBR322. So that also we would be studying in this particular lecture. So without further ado, let us start how this PBR322 works and what is the concept behind this. So PBR322 plasmid. Now, let us first of all discuss about the salient features of a plasmid. Now, what are the characteristic feature or what are the salient features of a particular plasmid? Let us recall this. However, I have already made a video out of this, the characteristic feature of plasmid, but since we are starting a new plasmid, new vector, so we have to talk about the salient features. So let us recall this. So function of ideal plasmid vector, how would you recognize or how would you look at a ideal plasmid vector? Let us first of all discuss this. So what are the characteristic feature? So the minimum amount of DNA should be present inside the an ideal plasmid. What do you understand from minimum amount of DNA? Let us understand this. The length of the plasmid should be as little as possible. Why am I saying that? The length of the plasmid should be as little as possible because the larger is your plasmid, the smaller amount of gene of interest you would be able to integrate. And the smaller is the length of your plasmid, the larger amount of gene of interest you will be able to integrate. Let me just write it down for your understanding. So, if the length of vector is high, gene of interest but naturally would be low, and if the length of vector is lesser, you can increase the gene of interest size. Obviously, this is because the vectors will be introduced inside a bacterial cell, and the bacterial cell has a particular size. If you exceed, or if the size of the vector itself is larger, you would not be able to introduce a bigger amount of gene of interest because of the limitation of the space inside bacteria. So this is the concept. So there should be a minimum amount of DNA present inside the plasmid vector. It should be less than 10 kb in size. Now let us talk about the relaxed replication control. Now, what do you mean by relaxed replication control? Now, this is a concept that it simply means that the replication of the plasmid should be independent of the replication mechanism of the chromosome. That means chromosome should be having a different mechanism of replication, and the plasmid that we are talking about, in which we have integrated our gene of interest, that should have a different replication mechanism, or it should be independent upon the chromosome, because it's not necessary always that the chromosome will be replicating. So there might be a condition where the bacteria is in stress, so in that condition the chromosome might not be replicating. So in understanding that, I would personally want my gene of interest to be replicating, to be amplified as much as possible, irrespective of what happens with the chromosomal material inside the bacteria. So that is why we are saying that a relaxed replication control is necessary. Again, in other terms, relaxed replication control simply means that a replication mechanism that is independent of the chromosomal replication. The plasmid will have its own replication mechanism, and that is primarily because of the presence of origin of replication, ORI region. Now, all of you would be knowing about the ORI region. ORI is origin of replication. Origin of replication. Now, what is origin of replication? It is a site at which, or it is the site from which the replication begins inside a vector. So in PBR322 also there will be an origin of replication that we would be talking about in the upcoming slides. So this was about relaxed replication control. Next is at least two selectable markers should be present. Now what do you mean by selectable markers? Now, there are two concepts over here, selectable and screenable. What do you understand from both of them? Selection means basically you will have to identify that whether your bacteria is transformed or non-transformed, whether you will have to differentiate between the transformed and non-transformed cells. So that is one of the prime requirement of selectable markers. That is why we use selectable markers. And when we are talking about screenable markers, then through the screenable markers, we are able to differentiate between the transformed bacteria that is transformed with the recombinant DNA and the transformed bacteria that is just transformed because of the vector. So selectable and screenable markers, we would be talking about them in the upcoming basically slides. However, in PBR322, we would be talking only about the selectable markers. There are other screenable markers in other type of vectors where we talk about the LAC-Z gene. So those are some of the screenable markers. We will be talking more about that in the next lectures. Next, only one unique recognition site. Now, what do you understand from this? Unique recognition site. Now, you might be knowing that vectors basically have unique sites on which or at which the restriction enzymes will be cleaving. Now, once the restriction enzyme cleaves at that site, you can introduce your gene of interest in that particular site, right? So suppose, for example, I'm saying that this is my gene of interest. Suppose, let us say that this is my vector. It is in continuation, right? Let me just use this. So this is my complete, let us say this is my complete vector. Now what will I do? I will use a restriction enzyme and I will cleave at this spot. Now, because of the use of the restriction enzyme, the two DNA fragments will be separated like this. Now, once you have separated them, what you can do is you can ligate your gene of interest, or you bring your gene of interest like this, and like this, you can integrate your gene of interest over here. You can integrate your gene of interest, like my finger is the gene of interest, and then this is the vector form, this is also the vector, and in between my finger is there representing the gene of interest. So, yes. What I want you to understand over here is that only one unique recognition site should be present. By unique recognition site, I simply mean that the restriction enzyme, at least, I mean, one restrictions, there can be multiple restriction sites. That means multiple restriction enzymes can cleave at different places, but an important feature is that one restriction enzyme should have one site only. That means if I'm talking about EcoR1 should cut at one spot only. It should not be cutting at multiple spots. That is what I'm talking about when I say the unique recognition site. Next, when you're talking about the selection process, the recognition site present in any of the two markers. Now, for the purpose of selection, now, as I've said that in the process of selection, what you do is you identify which is the transformed cell and which is the non-transformed cell. So, restriction site present in any of the two markers. So over here, I will be showing you in just a while that there will be two markers that are basically present over here. Two antibiotic resistant genes are present. Basically, I would be showing it to you in just a bit, and then we can discuss further on this particular point. So I hope you would have understood this slide. Just take a photograph of this so that we can move further. Okay, so I believe you would have taken the photograph. Let me just move on to the next slide. Now, talking about the PBR322 gene, what is the meaning of PBR322? Let us first discuss about this. So PBR322 in general, basically, if I talk about the full form of PBR, P means plasmid, B means Bolivar, and R means Rodriguez. So PBR322 basically is named on the two scientists who discovered it, Bolivar and Rodriguez. This is the number, basically 322 is the number at which this particular plasmid was obtained. There would have been numerous plasmids that they would have made, but this worked out, PBR322 worked out, and that is why this name has been given. Now, It is a very popular vector, let us say that, and it is widely used in the molecular biology laboratories throughout the world. Next, what is the size? See, generally these type of questions also are asked in the examination, that the size of this vector is 4363 base pairs, or you can call it as 4.3 kb is the size of this particular vector. Now, replication module of E. coli plasmid called E1. Now, what our replication module over here means, that what kind of replication is being followed in this plasmid. Basically, the replication module of call E1 is being followed over here. Now, what is call E1? Call E1 is basically a type of plasmid, and this plasmid is basically providing resistance against colicine. Colicine is basically a toxin that is secreted, that is produced by E. coli. So, if a particular bacteria is having a col plasmid inside it, then definitely what will it do? It will provide resistance against the colicin, and also it will produce colicin, and it will provide resistance against the colicin secreted by other bacteria. So, the replication module of this plasmid is followed. It permits, now what is the scenario, or why we are paying so much of attention over this? The replication module of E. coli plasmid is very what you say, important, because it will permit replication even under starvation or presence of chloramphenicol. Now you know that chloramphenicol is a very potent antibiotic. Now, as I already said, that it does not matter in case of pBR322. In case of an ideal vector, the replication control should be relaxed. That means it should not depend upon the chromosomal replication. In this scenario, pBR322, also the same kind of scenario is occurring, because it is following the replication module of call E1. In this also, the replication of the plasmid will be continuing without whatever impact is happening on the chromosomal thing. Even if the chloramphenicol is present, then what will happen? The bacteria would be under starvation. It would not face an ideal situation. Then also, the replication of this particular vector or plasmid will occur. That is the importance of the replication module of E. coli plasmid, or called E1. Now, I hope this is clear in your mind. You can just take a photograph of this and then we can move forward. Okay. Now, let us move further. So, 1 liter of bacterial culture. Now, let me just focus upon this particular point, that why is this so much of importance, or why the replication module of call E1 is so much important? Because it is providing us that 1 liter of bacterial culture would provide 1 mg of plasmid, even in the what do you say, the starvation condition. Even if the bacteria is under starvation condition, then also 1 liter of bacterial culture would lead to the production of 1 milligram of plasmid. Now, this is an important thing. It has been asked in several life science examination, that what are the selectable markers present inside the pBR322. So the different selectable markers that are present are tetracycline resistance and ampicillin resistant marker. That means these two genes will be present in the genome of the vector, and these two genes will be serving as the selection or selectable markers. We would be talking how these genes, talking about that in the subsequent slides, that how these genes will be functioning. Next is unique restriction sites for 12 different restriction enzymes. Now, as I have already said, that different types of restriction enzymes would be able to cleave, or would be cleaving at different spots on the vector, right? So there are how many enzymes? There are 12 different restriction enzymes. I will be showing you the diagrammatic representation in just a bit, so as to make you understand that which restriction enzymes are cleaving at which particular place. Now, sequences derived originally from 3 different plasmids. Now, let me just introduce this to you, that the pBR322 plasmid is just like an assembled computer, right? So in an assembled PC, what do? You bring, let us say, hard drive from a particular company, you take motherboard from a particular company, you take monitor from a different company. So this is what you do. You bring different components of nice companies, good companies, and then you assemble it at one particular point. The same thing is being done in PBR322 also. Remember, PBR322 is not a natural plasmid, it is an artificial plasmid. Now, in all the cases of an artificial plasmid, what happens is that you have to bring in different portions from different other plasmids and you have to integrate it at one place. So the same thing is being done over here. So there are three plasmids from which the different portions are taken up. So you can see over here BLAR gene, this, next is TET gene, and next is ORI gene. So these three genes are taken from different places. Where are they taken from? Let us talk about this. So the first thing that you can see is it is taken from the plasmid R1. Now what is R1 plasmid? R1 plasmid is actually the antibiotic resistant plasmid. Okay, that means it is a type of plasmid that would be providing resistance against a particular plasmid, resistance against a particular antibiotic. So BLRA gene is obtained from the R1 plasmid. Similarly, TET gene is obtained from R65. This is also an antibiotic resistant plasmid. Next is ORI gene. ORI, I have already told you about this, origin of replication, and it is obtained from PMB1. It is a close relative of plasmid call E1. Now I have already told you that the replication module will be following the call E1 plasmid. This is just because the origin of replication gene is coming from the PMB1, that is a close relative of call E1 type plasmid. So these are the three genes, remember, BLAR gene, TET gene, and ORI gene. The above two genes are basically coming from antibiotic resistance plasmid, and the third gene would be coming from the origin of replication coming from PMB1. Now, let us move forward and talk about the structural features of PBR322. Now, this is the diagrammatic representation of PBR322, and this is how basically it looks like. Now, if you can see, these are the restriction enzymes that are present over here, and these are the numbers at which base pair it would be cleaving. So you can see over here that HIN3, EcoR5, BAM-H1, SAL-E1, NDE1, PST1, PVU1. Basically, these are the different types of restriction enzymes that would be cleaving at different spots, at different positions in the PBR322 genome. Now you can see that I have already told you, let me just take a pen over here. I have already told you that this is the origin of replication. These are the two genes that are playing important role. For example, ampicillin resistance gene, this is one gene, and this is the this is the tetracycline resistance gene. So these are the two genes, ampicillin resistant and tetracycline resistance gene. These two genes are basically functioning as the selectable markers over here, and in these selectable markers, you can see these selectable markers, there are multiple restriction enzymes that are cutting at different places. For example, if you understand over here, SAL-E1, BAM-H1, EcoR5 is basically cutting in tetracycline resistance gene. And if I'm talking about ampicillin resistance gene, over here PVU1, PST1, they are cutting. And this is the entire length of the 4361 is basically the entire length of this particular plasmid. Now this is what I wanted to tell you, that there are two, that is why again and again I was saying that the selectable markers, there are two selectable markers over here, tetracycline resistance and ampicillin resistance. These were the two markers, and this is the basically structure that I wanted to show you. Now let us see what is written over here. So if you see, restriction sites are present within the markers. As I said, these are the restriction sites, right? These are present inside the markers, within the markers. What are the markers over here? Tetracycline resistance and ampicillin resistance markers. These are present inside or within the markers. And basically tetracycline resistance and ampicillin resistance, they allow easy selection. Now I've already told you that the function of these markers is selection of transformed from non-transformed, or differentiating transformed from non-transformed. Now DNA fragment inserted into the plasmid using PST1 and PVU1. Now, let us understand this, that the DNA plasmid that you want to insert, or let us say the gene of DNA fragment, rather, gene of interest that you want to insert, you can cleave this particular region by any of these restriction enzymes. So you can cleave this region by PST1 or PBU1 and then insert your gene of interest inside it. What do I want to say over here? Something like this. Suppose this is your ampicillin resistance gene, and I want to insert my gene of interest. Now let us say this is my gene of interest. So what will I do is I will cut this fragment with a restriction enzyme with whatever restriction I want to cut. Let us say BAM-H1 I am using to cut. Restriction enzyme will be used to cut, and the gene of interest like this and like this. So this is your gene of interest and this is your vector. This is the way in which it will be attached. So the same thing is applied over here. This is what is written: DNA fragment inserted into the plasmid using PST1 and PVU1. So in general, what is done is either you can use PST1 and PVU1 to cut and integrate into the ampicillin resistance gene. So this is the ampicillin resistance gene. These are the restriction enzymes that can be used to integrate. Now, if you are using them, then the gene will be inserted into the ampicillin resistant marker, right? Next, bacterial cells containing the recombinant pBR322, that means when the gene of interest is inserted in between this, what will you get? You will be getting a recombinant pBR322. That means it will be containing your gene of interest. Apart from that, the vector will also be present. Now, in this, if you consider this the recombinant vector that will be having your gene of interest over here, in this case ampicillin will be basically insertionally inactivated. That means over here your gene of interest would also be present. For example, this was the entire ampicillin resistant marker. Over here, or in this particular scenario, it was functional. But when you are inserting your gene of interest like this, let us say gene of interest has been inserted in between, what is wrong? Now the ampicillin resistance gene is not functioning properly because it has been separated by gene of interest. Because of this, now this ampicillin would not be functioning properly, but tetracycline would be functioning properly. Now if you are allowing this particular cell that is having this vector to grow inside ampicillin, it will definitely be killed. That means in the presence of ampicillin, it will be dead, and in the presence of tetracycline, it will be alive. Why? Because tetracycline gene is still intact. If you have cut it down with PST1 or PBU1, then of course the gene would be, this ampicillin resistance gene will be non-functional, but again tetracycline gene would be functional. That is why it will be surviving in the presence of tetracycline gene, but it will be surviving in the presence of tetracycline antibiotic, and it will be killed in the presence of ampicillin antibiotic. So I hope you would have got this clear in your mind. You can take a photograph of this. Okay, now let us move forward. Now, if I want to add my gene of interest onto if I want to create a recombinant PBR322, the previous one, I'm just discussing the previous thing only. So in this case, ampicillin, if you are putting your recombinant PBR322, this is the recombinant PBR322, if you are putting in ampicillin, it will be killed, and if you are putting in tetracycline, it will be surviving since it is intact. Now, if you want to create a recombinant pBR322 by integrating your gene of interest inside the tetracycline gene, of course tetracycline gene will be interrupted or insertionally inactivated. Let me just write it down for you. Insertionally inactivated. And because of this, what will happen? If you are allowing it to grow in ampicillin, it will survive. If you are allowing it to grow in tetracycline, it will be killed. Now, if BAM-H1 or SAL1 is used, now in this case, generally what happens is, if you want to integrate it inside the tetracycline resistance gene, you will be cutting it with BAM-H1 or SAL1. Now, insertional inactivation, I had already written over it. So this is complete. So you can take a photograph of this and then we can move forward. Okay, I believe you would have taken it. Now, let us move forward and discuss about the steps how this PBR322 is used in genetic engineering. So, let me just write it down over here. So, using genetic engineering, use in genetic engineering. So, how would you use this in genetic engineering? So, first thing first is that you will have to make the recombinant DNA fragment. How would you want to make a recombinant DNA fragment? Firstly, either I cut it with BAM-H1 or I cut it with SAL1 or I cut it with PVU1 or PST1. I can use any one of them. If I'm using BAM-H1 or SAL1, BAM-H1 or SAL1, then I will have to integrate my gene of interest in the tetracycline resistance gene. The gene of interest would be added into the tetracycline resistance gene. But if I'm using PVU1 and PST1, PVU1, PST1, then I will be integrating my gene of interest into the ampicillin ampicillin resistance gene. In both the cases, if I'm doing this tetracycline resistance gene will be now inactivated, and in this case, ampicillin resistance gene will become inactivated. Suffice to say that in both these situations, what you have to remember that you have to cut the gene of interest as well as the vector both with the same type of restriction enzyme. That means if you are using BAM-H1 or let us say if you are using BAM-H1, you have to cut out your gene of interest also with BAM-H1, and you have to cut out your vector also with BAM-H1 so as to produce the sticky ends, and in this way the gene of interest will directly be integrated inside the vector. That is the reason why you have to use the same kind of markers. So in this way you have created a recombinant DNA fragment. Now, okay. Now let us move forward. If you have created a recombinant DNA fragment, let us move forward and discuss the next thing. So this is the diagrammatic representation which is actually telling you how you can integrate your gene of interest inside the different selectable markers. Selectable markers. Over here you can see that DNA insert has been integrated inside the basically this ampicillin resistance, and over here you can see the gene of interest integrated inside the tetracycline resistance. So ampicillin has been inactivated over here and tetracycline has been inactivated over here because of insertional inactivation. I hope this is clear to you. Now let us move forward and discuss about the next process. The next process, as you can see, it is called as transformation. What do you understand from transformation? Transformation simply means that when you want to, this is your bacteria, okay? This is, let us say, your bacteria, and you want to put in your vector. And what was my vector over here? It was PBR322. I want to integrate my vector PBR322 inside my bacteria. This is what is my motto. Now, remember that the PBR322 can be of two types over here. That means it can be either recombinant PBR322, or it can be normal PBR322, because you are never sure in biology. The processes are not 100%. You will never be sure that the gene of interest that you wanted to integrate inside the vector was actually integrated or not. It might have been integrated. If it was integrated, then it will be called as recombinant PBR322. So let me just write it down over here. So when the gene of interest was integrated into PBR322, then it will be called as recombinant PBR322. And when gene of interest was absent, it will be called as normal PBR322. So this was the scenario. Both of these will be incubated in the presence of bacteria, and they will be allowed to enter inside the bacteria. This process is called as transformation. Now, let us move forward and let us just move to the next slide. Now, let us talk about the selection process. How would you do the selection? Now, as I've already told you that there are two markers that are selectable markers. There are two markers. What are the two markers? The first marker is ampicillin and the second marker is tetracycline. Okay. Now, both of these markers would be used to select the transformed from the non-transformed. How would we do that? Let us quickly see this. Now, there will be three scenarios that we are dealing with. Okay. Let us say that this is, let us say, bacteria number 1. Let us say that this is bacteria number 2, and let us say that this is over here. Let me just draw it over here. This is bacteria number 3. These are the three bacteria that we are looking at. Now, there will be three scenarios. Why did I say there are three scenarios? Now, let us say that the first bacteria that we are using was not at all transformed. It did not get transformed. So, that means when I am saying it will not get transformed, I simply mean that the vector will not be entering inside this bacteria. The vector could not enter. So we can say that it is non-transformed. Non-transformed. What about this? The second one. This second one has been transformed, and it is transformed with recombinant pBR322. Recombinant pBR322. The third one has been transformed with normal pBR322. Normal pBR322, right? So if this is non-transformed, obviously, if this particular bacteria does not have any gene related to tetracycline resistance and it does not have any gene related to ampicillin resistance. If I'm talking about transformed pBR322, let us say that it has been transformed at the tetracycline resistance gene. Let us say that it has been transformed at the tetracycline resistance gene. Okay. Now, and tetracycline resistance gene, and obviously if it is transformed at tetracycline resistance gene, so what is the characteristic feature? Tetracycline resistance gene is absent, ampicillin resistance gene is functional. I hope you remember why is this. It is the gene of interest has been added to the tetracycline resistance gene. Remember. What about this pBR322? In this, the tetracycline resistance gene is also present, the ampicillin resistance gene is also present. Both the things are present. Now, all these are now transferred onto a Petri plate. This is a Petri plate, Petri dish. And what I have done is, on this Petri plate, I have allowed, or I am putting antibiotic. And let us say the antibiotic that is present over here is ampicillin antibiotic is present. Obviously, since you are using ampicillin antibiotic, what will happen? This particular bacteria does not have either tetracycline or ampicillin resistance gene. It will be eliminated. In this way, you have differentiated between, these were the two transformed cells, and this was non-transformed. So you have differentiated transformed with non-transformed in this particular way by using one of the antibiotic resistance gene. So this was selection. Now let us move on. You can take a photograph of this. Okay, now let us move on to the next process, that is screening. In this screening, what will I do? I am left with only two bacteria now, and these bacteria are the transformed bacteria. Let us draw it like this. So this one is the transformed, and this is transformed through recombinant pBR322, and I am left with another bacteria that is also transformed, but it is transformed by only pBR322. Just for your purposes, I am just writing down that over here tetracycline resistance gene is absent, ampicillin resistance gene is present, and talking about this, ampicillin and tetracycline resistance gene both are present. Now, in the next step, what I will do is I will be transferring these two bacteria onto two different plates. I will be transferring these two bacteria on two different plates. Let us say that this is the master plate. In the master plate, I am putting no antibiotic. There is no antibiotic in this master plate. Now, if you are not putting any antibiotic in the master plate, what will it mean? That all the different types of bacteria will start growing over here. All the different types of bacteria will start growing over here. But let us, okay, just a moment. Let me just draw it a different color. All the different types of bacteria will be starting to grow over here. But in the next plate, let us say that I used ampicillin. Yes, basically, let me just write it. Just clear it off. Not ampicillin. I would be using tetracycline. Let me see. I am using tetracycline antibiotic. Now, if you are using tetracycline antibiotic, of course, the recombinant pBR322 will not be present because tetracycline resistance gene is absent over here and only ampicillin resistance gene is present. So ultimately, what will happen? It will get killed. Tetracycline resistance recombinant pBR322 will get killed over here in this particular case, and all the other, that means the vectors that will, or the bacteria that will be having pBR322, they will be surviving and recombinant pBR322 will be killed. So that means, let me see, let me just draw it like this. So let us say that the recombinant or the PBR322, these bacteria are surviving over here and the green ones are not surviving. Of course, I have just given you the color code. It is not about color. Basically, what we do is we do replica plating mechanism. In replica plating mechanism, depending upon the same location, we determine that which of these are surviving over here but are not surviving over here. So those that are surviving over here and are not surviving over here would be actually the recombinant bacteria. So that would be chosen, or those bacteria would be chosen, and those that are surviving, actually they are actually these one, pBR322 non-recombinant transformed bacteria. I hope this is clear with you. So now in the next, you can take a photograph of this and then we can move forward. Okay, so I hope this is clear. Let us move forward. Now in the next case, what you do is by doing the replica plating experiment, through the replica plating experiment, you find out that which one is the PBR, recombinant PBR322, sorry. You would be finding out which one is the recombinant PBR322, recombinant PBR322, and just transfer it to another test tube, let us say, and in this way the bacteria will be starting to grow and amplification. So this is the way in which it is used for the purpose of cloning. I hope you got a clear idea. Now you can take a photograph of this and then we can move forward. Okay, now let us talk about the advantages that this PBR322 has. It is a small size vector, so obviously it is easy to purify, and manipulation is also easy. You can make changes, you can easily purify it, because the smaller is the size, it is easy to purify, of course. Now easy selection of recombinant DNA. You can easily select which DNA is or which bacteria is recombinant because it is smaller in size. High copy number. What do you mean by this? The vectors that are smaller in size can multiply in a larger number. That means high copy number. So when I say larger number of vectors can be present because of replication, I simply mean that a large number of plasmids would be occurring, and in other words, it is called as high copy number. So it has been seen that around 15 copies per cell have also been seen. Amplification up to 1000 to 3000 when protein synthesis is blocked. That means when the protein synthesis is not occurring, the amplification level has been reached up to here. The Nick bomb, basically what does this mean? Bomb means basis of mobility. What do I mean to say? That means if a plasmid, let us say a F plus plasmid is present inside the bacteria apart from this PBR322, F plus plasmid is present. Now, if F plus plasmid is present, you would be knowing that it is a conjugative plasmid. If it is a conjugative plasmid, it will definitely allow the Nick bomb or plasmids like this that are non-conjugative, because this Nick bomb is present inside the genome. So it will allow the transfer or mobility of plasmids like pBR322 also, which actually are non-mobile or not, they cannot show motility. But if a F plus plasmid, something like that, that is conjugative in nature, if it is present, these plasmids can also move. This is what is meant. Now let's move. So this I have already explained it to you. Basically, it is referring to the mobility. Now let us move on to the next slide. Now let us talk about some of the PYQs or previous year questions. Not necessarily PYQs. These questions are also I have taken from different internet resources. Now let us quickly do this. So the first question is, two antibiotic resistance gene on vector pBR322 are basically what are these two different genes that are antibiotic resistance, basically ampicillin and tetracycline. You already know that, so the correct option is 3. Let us move on to the next slide. Now, what is pBR322? I am hopeful that now you will be knowing about this. So this is a plasmid vector. Correct option is the fourth one. What is the term for the process of introducing foreign DNA into pBR322, thereby creating a recombinant plasmid? What is the term for the process of introducing foreign DNA into pBR322? That means now this was, let us say, a pBR322. You are cutting this gene by, let us say, BAMH1, and then you are putting your gene of interest like this. This is your gene of interest. So what is this process called? This process is called as ligation. This process is called as ligation. That means you are ligating your gene of interest inside your what do you say, plasmid. This is called as ligation. So I hope this is clear. Now let us move on to the next slide. What is the purpose of tetracycline resistance gene in pBR322? Obviously, it is basically it serves as a second antibiotic resistance marker, because tetracycline, apart from ampicillin, so ampicillin is the first one, and apart from the ampicillin, the tetracycline resistance gene is also present, so it is serving as a second antibiotic resistance gene. Next, which enzyme is commonly used to cut DNA at specific sites for the purpose of inserting foreign DNA into pBR322? So which enzymes are used? Obviously, restriction endonucleases enzymes are used, because BAMH1 and PST1 all are these restriction enzymes only. Next, what is the name of the process used to transfer the recombinant plasmid with foreign DNA into bacterial cells? Now, we've already talked about this. The process through which we are transferring the vector inside the bacterial cell is called as transformation process. Transformation. Next, which of the following is not a typical use of PBR322 in molecular biology research? Remember, basically, it is not at all related to protein purification. It is not an expression vector. It is a cloning vector. So obviously, protein purification is not associated with PBR322. I hope this is clear to you. Okay, now, so that was all from my side. I hope you enjoyed this particular lecture. Do tell me how did you like this lecture, and I would be making more videos on vectors in English as well. So please stay tuned, and also tell me in the comment section which other videos you would like to take me. We would like to you would ask me to teach, so that I can make those videos. Thank you so much. Have a good day.